I am very happy to introduce today's speaker, Professor Jeffrey Schell, who is now the Henry and Grace Doherty Professor and Chief Scientist, Chair, Head Boat Guy at Sea, which is the Sea Education Association out of Woods Hole. He's been there for a long time. I couldn't tell you what he's been doing for the last 25 years, but I could tell you what he was doing last millennium when he got his master's degree here at the Marine Sciences Research Center. When he was young and good looking, like many of you, not all of you. So Jeffrey cut his teeth here, safe to say.
Yeah.
Had a very impactful time here, safe to say.
It was important. This is important.
Did a lot of crazy things that we can talk about at lunch, safe to say. After getting
allowed to anymore.
Oh no, we're gonna get into that. Got his master's in 96-ish. Last millennium, doesn't really matter. Then he went to get his PhD from the University of Wisconsin later on. He's been with Sea since what, 2004 or something like that. He's a professor, 2003 as a professor, and he still goes to sea 4 months a year. His passion for education and marine education is unparalleled. I mean, this guy's got some stories for sure that you'll want to listen to. Also, I want to point out, since this is Alumni Day, you didn't know this, Andy Matthews, who graduated with Jeff Schell right around the same millennium, is in the back there. Andy got his master's also in from marine sciences. He still lives here. He's the head of math, science, and technology at the Mount Sinai School District. Longtime biology teacher. Also winningest coach of the Bay Scallop Ocean Sciences Bowl. He's like 8-3. I don't know how he does it. He probably cheats, but you know, so did Belichick. So he's right there with Bill. So Andy's here, and he's going to be joining us for lunch too. If you want to talk to someone who was here back in the day, and also, you know, for those of you who are kind of interested in what you may be able to do with a degree beyond science and stuff like that, both these guys have stories. I can tell you about a lot of stories about what we used to do, or what they used to do back in the day before cell phones. So with that, I don't know what he's going to talk about. Something about water plants or something. I don't know. And he's very hands-on. He's got props. So I'm very much looking forward to what he has to say. He's going to talk about sargassum. For those of you in the Caribbean who've ever been down there, sargassum is a major problem now. And we were down in Barbados last summer, and it's a big deal. So it's a very timely thing. So Jeff,
all right, thank you, Zach.
Take it away.
Before we begin, I don't know if you knew this, pop quiz, since this is the last seminar. Pass around this sheet of paper, please fill it out. I'm going to give you a few moments to put your answer on this sheet of paper and then pass it forward. Oh, I'm going to. So you'll need a pen. If you want your name on the publication, then include your name on your answer sheet here. So where is the Sargasso Sea? That will be one question that we will attempt to answer. I've been trying to answer this question my whole career.
No Googling.
Where is the Sargasso Sea? No, yeah, just what you know. Don't look it up. What you look up online is probably going to
Bob Oller's cheating.
Anyway.
Look at
Yeah.
the answer.
Please do your own work. Please do your own work. For this. So I'll give a little bit of my history. John gave a little bit of it, but I'll give a little bit more while you're having time to pass that sheet back and put your answer on the paper there. Yeah, pass it on forward. But yeah, I started in Ohio and didn't see the ocean for the first time until I was in high school. But that was enough for me to then pick the direction for my career. I didn't know what it meant to be an oceanographer. I didn't even really know that term at the time. I was like, I'm going to be a marine scientist or a marine biologist. That's what I thought I was going to do. I didn't know how I was going to do it. And really, it was coming here after my undergraduate time at Holy Cross. It was coming here where I actually learned, like, what does it mean to be a marine scientist or an oceanographer? I really didn't have any clue, even though I was lucky enough to be accepted to come here as a master's student. And I was lucky enough to work with Steve Morgan. He introduced me to what plankton were. introduced me to crab larvae and how they have these great adaptive behaviors and migration patterns and adaptive morphologies to protect themselves as they're trying to live their planktonic life and then make it back home as adults. So that was because of my time here at Marine the MRC. So really impactful time in my career and influenced the rest of my time as an oceanographer. I have continued to study plankton, and now I'm studying planktonic algae, sargassum. So we're going to dive into slides here. So some of the questions we're going to try to answer are: what is sargassum? Why should we care about it? Where is the Sargasso Sea? And we'll take a look at everyone's answers here. I'm really curious. All right, very great. Has sargassum biodiversity changed over the years? And we'll define what I mean by this. And what is influencing sargassum growth? And we already had sort of some foreshadowing, thanks to John here, about some unusual things that have been going on the last decade or so down in the Caribbean. And we're going to try to understand a little bit about sargassum growth and blooms. So sargassum is a brown algae in the order Cucales, worldwide global distribution of benthic sargassum attached to the seafloor, anywhere from the tropics to temperate latitudes, worldwide. But the sargassum that we'll focus on here today is holopelagic, always floating around the sea surface. It's never attached to the seafloor. And there's a few species of sargassum that are known to do that. And it's only in the North Atlantic Ocean where you find them. So we're focusing on that aspect of the genus Sargassum. And it's not just this great photosynthetic organism, but it's an entire habitat for a variety of organisms as well. So a number of organisms attach and grow on the sargassum itself: colonial bryozoans, solitary barnacles, and anemones, colonial hydroids. Those in turn then are the base of a food web for an entire ecosystem occurring in this floating habitat. Crabs, shrimp, fish, nudibranchs, as you can see, polychaetes, snails, flatworms, and more nudibranchs. So an entire ecosystem develops on this floating habitat. This is an endemic species of nudibranch found only in this floating ecosystem. So an entire food web develops on this floating habitat. And that in turn brings in a lot of migratory fauna as well. Large mahi-mahi, flying fish are known to lay their eggs on sargassum. And juvenile sea turtles and eels and seabirds are also known then to frequent these habitats. So what is sargassum? It's a habitat that's worth protecting. And it's why Sylvia Earle has identified the Sargasso Sea, if we can define where it is, as a place to protect because of this unique floating ecosystem. It is an area of special abundance and diversity of species. Unusual or representative species or habitats of ecosystems, it's clearly that. And that there are up to 10 species that are endemic, like that nudibranch I showed earlier, 10 endemic species to this floating ecosystem. And this was identified as the first hope spot of many that Sylvia Earle and others have identified as special, unique places that needed protection. A Bermuda effort called the Blue Halo and the Sargasso Sea Alliance, a community of nations that have come together to promote the protection of these high seas environments, including the Sargasso Sea as the first one. And floating sargassum, even in the United States, has been recognized as a critical habitat for many pelagic fisheries species in the southeast sector. So SEA has been part of the effort to draw attention to the ecosystem value of sargassum as a habitat by summarizing a lot of the data in our Newstone tows that we have conducted ever since 1971. So Sargasso Sea is a place of great beauty, ecological value, a golden floating rainforest at the surface of the North Atlantic, an oasis of marine life. So clearly, sargassum is an important habitat in the North Atlantic Ocean. So if we're going to protect it, we need to know where it is, and that leads us to our next question: where is the Sargasso Sea? So this is a question that we thought we kind of knew the answer for a long time. You can find it on maps shown lots of different places, but in 2011, Gower and King kind of upended our view and understanding of where sargassum is found. Is it found in the Sargasso Sea or not? And it was this paper that caught our attention at SEA because using a new technique, remote sensing satellite imagery, they had a really interesting hypothesis about where sargassum comes from. It doesn't come from the Sargasso Sea. It just ends up in the Sargasso Sea. So some of the things that they determined, their hypothesis is that the source of sargassum is actually the Gulf of Mexico, where it blooms in the spring, and these different colors here representing how sargassum moves through the system across the seasons. And we see it exit out of the Gulf of Mexico in the Gulf Stream, and then ends up in the Sargasso Sea. It doesn't start there. At least that was their hypothesis. And in their paper, they provide a nice little hypothesis that you could go out and test. Presumably, new growth is occurring in these areas. And you can see that then in the sargassum, nice light yellow, newly growing sargassum, moving on through the system, infected out into the Sargasso Sea because of the Gulf Stream. And then through the fall and winter months, it moves and then eventually dies out. They lose the signal, and they presume that it must have just aged and organisms grew on top of it, and then it sunk out, waiting until the next year's spring event to occur. And that went counter to the sort of 15 years at this point in time, the experience I had with sargassum as SEA sails its ships from Woods Hole down to the Caribbean and back year in and year out. So we wanted to go and test this hypothesis and argue against it, counter it with our own data set. I had a unique opportunity in 2012. It was going to be our 240th cruise sailing with SEA. And it was a cruise track from Key West going to St. Croix, and there was an opportunity to test this pathway of sargassum directly. And it was a fun opportunity to work with my students and get them excited about testing a real world hypothesis. We had this paper to counter. We were either going to validate or refute this hypothesis. So it got them all excited. And so what is the work that we do at SEA and work that we do with students? Well, we tow this Newstone net, 1 meter wide, 1 meter tall, 333 micrometer mesh. And let's take a closer look at the Newstone tow. So SEA has been towing this at least twice a day, every day since 1971. Anytime the ship is sailing, this is our sort of bread and butter oceanographic sampling, along with a whole suite of other oceanographic equipment. The Newstone tow, something that we do all the time. And SEA has been collecting data on sargassum, zooplankton, marine plastics, marine tar. So it's an incredible data set. So we have thousands upon thousands of Newstone tows worth of information and data to, one, address this hypothesis that was proposed, this Gower and King hypothesis, as well as this understanding the overall distribution of sargassum and how it's changed over the years. So just a beautiful view. Oh my gosh, don't you want to just go swimming right now, this cold day here in December? Oh, to be down in the tropics, swimming with sargassum. So a typical view of our Newtown tow, sailing and coming across windrows of sargassum. And this is a video taken during that cruise. And then when that Newtown net comes back on board, we rinse, sort, and mass by species the different types of sargassum that we find. We sort and identify and count all of the organisms that we find in that sargassum. Count all of the critters. And to test this hypothesis specifically, so this is a level of analysis that didn't begin until we wanted to directly address Gower and King's hypothesis, is we really started paying attention to the age and the color of the sargassum. This idea that there's new growth in the spring and that it would age by the time it got to the Sargasso Sea, so that it should take on a much darker brown color as it's reaching its end of its yearly cycle, and was supposed to presumably sink out once it got to the South Sargasso Sea. So we started paying a close attention to the sargassum like we had never done before. And this will be important for later on. So as we were leaving Key West and sailing, getting into the Florida current and then eventually into the Gulf Stream, we of course were seeing sargassum as expected. This is a time of year where Gower and King would have suggested we would still find it. And we were finding sargassum that looked fresh and new. We were finding sargassum that looked pretty old as well. So that wasn't quite matching up with their hypothesis. And as we were moving our way into the Sargasso Sea, the Bermuda Triangle, we were going to go right into the heart of it. And we got all the students excited for all the myths of the Sargasso Sea. And there were beautiful days like this. But then it's not uncommon to have days like this where easily 24-foot seas. So height of eye, this is 13 feet above water. And then if you're standing, that's another 6 feet. And if you can't see the horizon, you know you're over 20 feet of water coming over the rail. So at this point, we're not towing the Newtown net. The sargassum is just coming on board. So that's great. So as we made our way across the Sargasso Sea, we had days like both of those. Beautiful calm days, rough seas. But more importantly, we continued to see sargassum. We started seeing more of it. But again, it was a mix of young and old sargassum, not quite matching with the Gower and King hypothesis. As we continued south, we continued to see more sargassum, and this now was surprising even in our historic data sets. We didn't expect to see so much. And again, it was a mix of young and old. Again, not quite matching the Gower and King hypothesis. And we thought, in at least all the years I had been sailing these trips, once you go south of 20 degrees, you should stop seeing sargassum. Right? We're leaving the Sargasso Sea. And it seems like that matches with a lot of people's expectations of where the Sargasso Sea is. Right? That area bounded by the North Atlantic Gyre. A sea without a shore, right? The currents represent the boundaries. So we should be leaving the boundaries of the Sargasso Sea at this point. And in Gower and King's satellite data, they didn't see any sargassum past this mark either. They thought everything sunk out and died in the South Sargasso Sea. Well, this is 2012. That time period is important. Because as we crossed that line, we started seeing more sargassum than SCA had ever recorded up to that point in time. So a massive amount of sargassum. And as we returned to shore, we started seeing these headlines throughout the rest of that summer of 2012. Changes in the air, seaweed everywhere. Fisheries Division response to sargassum invasion. Where's the beach? It's under the seaweed. John was just talking about it as he was flying over the beaches of Barbados. Piles and piles of sargassum washing up on the beaches. Incredible economic devastation in these islands when this sargassum is washing on shore. And this was, at the time, unprecedented. Some of the earliest questions were, well, what is this stuff anyways? They were just, in people's living memory in the Caribbean, they didn't know this was sargassum because sargassum never showed up there. I don't know if anyone in mapping where the Sargasso Sea is included the Caribbean. Some a little close, perhaps. But up until this point in time, you didn't find sargassum in the Caribbean. So this was surprising. And we thought perhaps it was a one-off event, but then these inundations occurred again in 2014 and 2015, and even bigger. And we now know 2017 and 18 had an even bigger bloom. And at this point, it has gotten the attention of a wide variety within the oceanographic community. And a paper by Wang and others in 2019, published in Science, maybe folks have seen this one, have identified and given a name to this phenomenon, the Great Atlantic Sargassum Belt. And that these blooms have periodically reoccurred and seem to be getting bigger and bigger with time. And I don't know if anyone had the Sargasso Sea that in this area here, but at this point, it seems like we have reached a tipping point, a new normal in the North Equatorial Atlantic for amount of sargassum. And the blooms continue. 2019, now we haven't even really had a pause. Now it seems like every year these sargassum blooms are coming through, forming off the west coast of Africa, following the trade winds and the equatorial current coming through the Caribbean and Gulf of Mexico. And what only seems to vary a little bit is how big is the bloom this year. There doesn't seem to be a cessation or a pause in between the arrival of sargassum anymore. So the earlier questions were, well, what is it? Well, we quickly realized it was sargassum, but then, hey, where is it coming from? Well, of course, it must have come from the Sargasso Sea, but we'll see if that's true or not. Why is it happening? We still haven't answered that question yet. Has it happened before? We'll take a look at that. And what can we do about it? A lot of people are working on that, trying to make lemonade out of lemons, so to speak. But one thing we could do at SEA is we look through SEA's archive of data and see, well, in times past, had sargassum ever made its way into the Caribbean? Can we get a sense of, has this ever happened before? And as I mentioned, we started that cruise trying to test Gower and King's hypothesis, and we'll address that in a moment. We ended in this really unusual event. We were right there at the start of that bloom, the first of now what we recognize as a recurring sense of bloom. But as we looked into SEA's archive of data, all of these Nusantos, a pattern revealed itself that we didn't even think to look for. So that will inform lots of the answers to questions to come. So at the time, we were able to summarize in the old paper data sheets that we had to go back to and enter and do all the quality checking, 6,610 tows across the range of areas that SEA sails. And we divided it up amongst a variety of regions, and we mapped where SEA had collected sargassum. And if I were to draw a map of where the Sargasso Sea is, based upon where sargassum is found, the areas we would define it are based upon percent occurrence of tows that found sargassum and the average amount of sargassum across this line here, which amounted to about 2 or 3 clumps of sargassum per Newston tow, as far as the total density. And this would have been the map, the circle I would have drawn around where the Sargasso Sea is, at least defined as by where do you find sargassum. At least 1 out of every 2 tows, if not much more, are you finding sargassum in our Newston net. So how does that match up where SEA's archive found sargassum in the Gower and King hypothesis? So it kind of overlays nicely, but we have to remember that this is a seasonal phenomenon. So if we look at our data just in the fall, these are our fall cruise tracks, and that matches up pretty nicely where they were seeing it in their satellite data. That's where our highest concentrations of sargassum were also seen. And where they weren't seeing it, we also were not seeing a lot of sargassum. In the spring, however, we did sample here. We did sample here and it matched up nicely. But for some reason, their satellite data wasn't picking up a pretty significant signature of sargassum that we were seeing in our Newston tows. So it's not capturing that signal for some reason. somehow the way the sargassum perhaps is distributed, it's not picking it up. And what also was not matching with the Gower-King hypothesis is, as we looked at this sargassum, there was no rhyme or reason to the pattern of age of sargassum. So there wasn't this cycle from the Gulf of Mexico to then old sargassum here in the south. We found healthy, growing, big clumps of sargassum just about anywhere we were sailing in these parts. But the sort of unexpected pattern that we observed, and something that I haven't really talked a lot about yet, is not all sargassum is the same. Historically, there are two known species of drifting holoplanktonic sargassum. Sargassum natans and Sargassum fluitans. And later on afterwards, if you want to come and take a closer look at some examples of them, I encourage you to do so. Sargassum natans is the one that has a little spike off of the end of the float, really narrow and long blades, where Sargassum fluitans has shorter, thicker blades, oblong to oval floats with no spine. And fluitans also has thorns visible along the stem or the stipe, and along the stipe of natans, you don't see any of those thorns. So morphologically very different. We have always been tracking those two species of sargassum. They were well understood, well known to be the two types of sargassum that you find out there. We compare the distribution of those two. This is just subtracting one from the other to get a sense of which one is more predominant in different geographic regions. And we see that Sargassum natans seems to be most predominant in the South Sargasso Sea. And we see fluitans spread sort of throughout. So we had to sort of reconcile this data to Gower-King's hypothesis and some of our other observations. And this was the model that we proposed, is that the Gulf of Mexico indeed is a source for sargassum, but for Sargassum fluitans. So this is probably the home range for Sargassum fluitans. It advex out of the Gulf of Mexico in the Gulf Stream and cycles somewhere to the East Atlantic. We don't sample over there, at least we hadn't yet at this point in time. And presumably some of this then can follow the North Atlantic gyre and make its way back into the Caribbean, sort of full circle, so to speak. And some of the large amounts of sargassum that we were getting in this Eastern Caribbean zone in this data set were in those last few years, 2012, those big events that occurred. And Sargassum natans, we think, is this home range is the Sargasso Sea traditionally. And this is why the idea of old versus young sargassum, that aspect of the Gower-King hypothesis wasn't matching up, because we had some sargassum that was just living its whole existence in this area here, young and old, with then Sargassum fluitans working its way through the system periodically. So sargassum seems to be distributed beyond its historic bounds of the Sargasso Sea. We saw that. Both of them did show a seasonal pattern going on. That was evident. So that matched Gower-King. The two species have overlapping but distinct ranges. That is important for later on. And this unusual event, where is that sargassum coming from? We thought there was something about perhaps Sargassum fluitans' path around the North Atlantic gyre that was going to help us understand where that sargassum came from that was coming into the Caribbean. So where is that sargassum coming from that was flooding into the Caribbean? We focused on historical and in situ observations. That's what we had access to at SEA. But in trying to answer this question, other researchers have used remote sensing and modeling approaches to try to answer that question. Where is all this sargassum coming from? But in the early stages, we were looking at our own data set, as well as looking at the historical record to try to understand where this sargassum came from. So if you go back to Clummers's logs, people have tried to estimate, looking at his logbooks, where he was at certain points in time And he does reference sargassum, and the first time he did so, we think was approximately there on the 16th of September, 1492. And he mentions in his logbooks 11 times out of the next 24 days, citing sargassum. So this is our first understanding of the distribution of sargassum in the North Atlantic. Note that it has a pretty far to the east signature of sargassum in his historic logbooks. Ben Franklin, actually in charting the Gulf Stream, not only based his observations on temperature and the distribution of whales, but he also talked about the distribution of Gulf wheat. So that's looking at another sort of historical look at where sargassum can be found. This is a map by Crummell in 1891, but what I want to point out first, sticking with the chronology here, is then in 1804, Alexander von Humboldt defined the Fucus vein to be in this region based upon shipboard observations. And again, note this big eastward side of the Atlantic, a source of sargassum. Matthew Fontaine, well, summarized by Matthew Fontaine Maurer in 1855, talked about Reynolds' observations of sargassum, again from shipboard observations. In Maurer in 1855, Physical Geography of the Sea, the father of oceanography, had this strong eastward component where he mapped the Sargasso Sea in his book. And then it was Crummell in now 1891, and we're starting to see a shift of sargassum from the east toward the west to more contemporary times. And we have Crummell's area shown here. We have Wing in 1923 mapped here. This is work summarized by Dooley, 1972. 1964, Conover and Seaberth suggests that the middle of the Sargasso Sea is here just south of Bermuda. So historically over time, it seems like there had been a shift of an eastward source of sargassum to a more westward North Atlantic positioning of sargassum. So we thought there must be something going on. The North Atlantic currents and winds have shifted. Sargassum distribution is just piling up over here on the eastern side of the North Atlantic like it did 100 years ago. So we thought if we could just get over to the East Atlantic, we would find where all this sargassum is coming from. And the reason we were interested in sort of diving into this historical literature is because we were trying to counter a different idea, a different hypothesis, this time based upon modelers' data rather than remote sensing. Johnson and others suggested, based upon their modeling data, is that the sargassum washing up on these beaches was actually coming from the equatorial Atlantic Ocean, a place that had never had any historic recordings of sargassum before. So based upon high-com modeling, they would just backtrack wherever sargassum washed up on the beach, based upon wind and tide or wind and currents known at the time. So we wanted to counter that hypothesis. So that was a pretty bold hypothesis, suggesting that the sargassum is coming from here, where there had been no historical precedent of that ever being a place for sargassum. So we thought for sure, but in the summer of 2014, one of our ships, the Corvette Kramer, did some transatlantic voyaging, and there was going to be an opportunity to test this hypothesis. We had a cruise that was going to sail from the Canary Islands to the Caribbean right at this time. And we were presumably going to sail right through this big patch of sargassum and show that the modelers didn't quite have it right. So I got my students all excited. We set sail from the Canary Islands, and I thought for sure when we crossed into Matthew Fontaine Maury's boundary, the sargassum was just going to show up. It was going to be great. So we set sail, and every day we'd come up on deck and look for the sargassum. We didn't see anything, and when we crossed Matthew Fontaine Maury's boundary, that's fine. Maybe Columbus had it right. So we got to that boundary. I was so excited to have my coffee, came up on deck and looked. No sargassum. All right. I'm a big fan of Humboldt, Alexander von Humboldt. Inspiration to me. I was like, for sure, he had it right. His Fucus band. So a few more days on, and I was like, all right, tomorrow, everybody, get ready. we're gonna see sargassum. We didn't see any sargassum, you know where this is going, right? And at this point, we've been 3 weeks at sea looking for sargassum and not finding any of it. And the captain at this point is like, we're supposed to be in port in 2.5 weeks. Like, we gotta go. We're not in the trade winds. So we had to give up the hunt, sadly. Our historic hypothesis of where the sargassum was coming from was clearly wrong. And yeah, to say I was heartbroken would be, true. Yes. So quite disappointed, and we sailed on and we were starting to catch the trade winds. And then on the 1st of December, everything changed. It started out as your typical thing. We were just getting ready for our morning science station. Hove 2, we started deploying equipment. CTE went over and came back up. We're drawing water. We're getting ready. It's like, okay, let's do our new stand tow. We got up to speed, put the net in the water. I even put a GoPro on it because it's like, I got to do something with my time. Like, I'm not processing sargassum. So let's put the GoPro on it. And up from aloft, I hear, sargassum! Oh my goodness, everybody came up on deck. And yes, we saw our first clump of sargassum after 3 weeks at sea. And not only did we see a clump, we saw a windrow that was probably as wide as this room from one horizon to the next horizon. And then as you looked further on, you saw another one of those windrows and another one of those windrows. And lo and behold, we were right in the middle of the 2014-2015 bloom of sargassum. It did not come from where I thought it was going to come from. It came from the equatorial Atlantic, just like the modelers predicted correctly. They were right, so I'm proud of them. So that's great.
Are you?
I am. But yeah, we're gonna get a view of what this bloom of sargassum looked like. So I had prepped the students, of course, at this point. We've been looking for sargassum for 3 weeks. And what we thought we were also going to find with this sargassum was this blooming ecosystem. Fish, seabirds, turtles, everything. We thought that would be a part of all of the sargassum, as I talked about earlier. This wonderful resource out here in the middle of this blue open ocean area. This oasis of life. So that's why I had the GoPro down underneath, and I was expecting to get this beautiful image of all of these fish swimming around. But oddly, we weren't seeing that. So that surprised me at first. So a couple things were going on. So after this first encounter, we saw sargassum every day. And more sargassum than in my living memory of sampling sargassum. We got to a point we came up with a new scientific measurement, the length of one of our scientists, of how much sargassum was in our net. So how many gabos of sargassum have we collected? So, but that wasn't the most surprising thing. It wasn't the amount of sargassum. It wasn't the lack of the rest of the ecosystem that we thought would associate with sargassum. It was the fact that this sargassum looked like no sargassum I had seen in my history of studying sargassum. This was something different. This is not sargassum natans 1 or sargassum fluitans 3 like I showed you before. This was something different and coming from a different part of the ocean where sargassum presumably doesn't come from. We have since identified this historically, going back to Albert Parr, who was the then director of American Natural Museum of History. He did some cruises and he was, like me, really excited about sargassum. Spent a lot of time looking at it. He described sargassum natans 1, the one who I introduced you earlier. He described sargassum fluitans 3, I introduced you earlier. But a rare one, and that's why it was given number 8 in his catalog of sargassum, because it was so rare, but he observed it and described it, sargassum natans 8. It was so rare that then people never saw it again and paid any particular attention to it. And why I say historically, it's always been presumed there are two species of sargassum. But we are now seeing this form of sargassum in huge abundance, coming from an unusual area. So we since went on and then reminded folks of Albert Parr's work and reacquainted people to Sargassum natans, because now it was one of the predominant types of sargassum you're seeing in the North Atlantic. And it was part of these large blooms inundating the Caribbean. And then we were struck with the question then, if we have this other type of sargassum coming from an unusual part of the Atlantic Ocean, what does that mean about the ecology of that system? And it was us trying to understand, why were we not seeing the large abundance of other marine life associated with all of this sargassum that we found here? So as we take a closer look at this sargassum, I'll point out just the different structures. When the sargassum is in the water, what kind of habitat does it provide to the variety of small organisms and then larger organisms that are typically associated with this drifting community? I could just get lost. I could just watch this like a screensaver all day. Oh my goodness.
Jeff, is this looking down?
This is looking down. And what's happening here is the sargassum is being, as our ship drifts back, it's being pushed underneath our ship and then mixed around. So sargassum doesn't normally always float in that sort of deep manner. We're going to start taking closer looks at each of the sargassum types. And this was the new one, or the rediscovered, previously rare form of sargassum, Sargassum natans. And notice, as you see that clump, you can almost like see right through it. The branching is very open structure. It's not very compact. And thus we hypothesize that it doesn't provide as much refuge for some of the smaller organisms. And then if you don't have those smaller organisms there, it's not an attractive habitat for some of the larger organisms. And thus maybe this explains why we are not seeing as rich of a marine ecosystem develop around this type of sargassum. We've since learned that the types of hydroids that are found on that type of sargassum are different than the hydroid species that you find on other sargassum. The Sargassum fluitans is one of the more historic, more abundant types of sargassum. Much more densely compact, branching. And thus our hypothesis is that it provides much greater refuge for the smaller organisms. And thus those smaller organisms attract larger organisms and then an overall richer environment. And we find that Sargassum fluitans typically has, at least in our sampling, a greater abundance of organisms, as well as a greater diversity of organisms living in this type of sargassum. And then we'll see that for Sargassum natans 1, much more compact than the other two. And then now we're seeing just some stills to get a sense of this. Just how the architecture is very different of the different types of sargassum. As we mapped the distribution, we had already seen how Sargassum natans 1 was home to the Sargasso Sea. Sargassum fluitans sourced out of the Gulf of Mexico and cycling through. And with Sargassum natans 8, even to this day, this is in 2015, but even to this day, we have not seen Sargassum natans 8 make its way and establish itself in the Sargasso Sea. So there must be some environmental tolerances that are separating out these species into these different areas. That'll be important later. But what's also important, at least at this time, is we were looking at SCA's historic data set. Of course, we're seeing the bloom in 2011, the bloom in 2014, this increase of sargassum in the Caribbean region that had not been there before. But what's important is that if we look across that same timeline, the amount of sargassum in the South Sargasso Sea has not shifted. So where is this sargassum coming from? Well, it's not just a movement out of the South Sargasso Sea into the tropical Atlantic. What's going on in the tropical Atlantic seems to be its own thing that now it's maintaining itself. So we didn't see any shift in abundance here across time, but we've seen this huge shift coming from here. So again, this supports the modelers, supports the idea that the source of this sargassum is indeed the tropical Atlantic. So now at this point where we have tons of information, we know that answer. But at the time, we were still trying to figure out where is this sargassum coming from. So this helped to support the idea of this tropical Atlantic source of these large blooms. So Sargassum natanzate indeed coming from that region. Our historical hypothesis wasn't supported, but other questions arise. And of course, I've foreshadowed many of these. We are interested in looking at the ecological value of these as different habitats. And that, of course, requires a different technique. So no longer the Newstown net, which just mixes all the organisms together. We needed to get one clump at a time, look at the organisms that you find there. So the highly technical dip net became our tool of choice. And just bringing one cup of sargassum aboard at a time, and then measuring everything you can find on it in as much detail as possible, so that you could then start answering a simple question: does one type of sargassum harbor more and a greater diversity of organisms? And I've already foreshadowed it. Yes, for crabs, if you compare Sargassum fluitans and Sargassum natanzate, Sargassum fluitans is a greater abundance of crabs. Similarly, it has a greater abundance of shrimp. We see that same pattern hold up with amphipods. And overall, Sargassum fluitans in our initial study, just as we were sailing across the Atlantic and making these first observations, 14 species present across our Sargassum fluitan samples, and only 9 species found across our Sargassum natan samples, and of a lower overall abundance as well. So greater variety of shrimp, amphipods, isopods, flatworms is fine on Sargassum fluitans 3. That was an initial study, and then we were able to follow up that study over the years as we got more and more samples to ask this question across greater regions. So is that a phenomenon just occurring in the tropical Atlantic, or do we see these differences play out anywhere we find this sargassum? And indeed, across all 3 types of sargassum, across the entire range that you find sargassum, you end up seeing a greater abundance and taxa richness on Sargassum fluitans 3 compared to the other 2. And within that, a different community of organisms. For example, at least in this study, we only found that endemic species of nudibranch only on Sargassum natans 1 in the Sargasso Sea. So the community that you find depends upon the kind of sargassum that you're looking at. But the biggest question that everybody wanted to answer was, well, why are these blooms occurring in the first place? And that's where the growth of sargassum comes into play. So I always ask my students to turn in those why questions into what questions. What are the factors responsible for the development of the Great Atlantic Sargassum Belt? And can we try to understand why these blooms seem to fluctuate over time? Why is there a cessation? Why are they seem to be getting bigger and bigger over the years? And there's some information that we have available to us at SCA that others didn't have, is that not only are we seeing this shift in abundance over time, but we also see a difference in the composition or the character of these blooms. So just looking at a subset of SCA's data set here, only about 660 toes, we see the composition of these blooms also seems to shift early on. Early on, fluitans was actually the predominant type of sargassum in these early blooms. Then we came across that rare species, Sargassum natans 8, in the bloom in 2014 and 15. Now, as the blooms seem to be getting even bigger, seems to be supporting all 3 types of sargassum in nearly equal amounts, thus supporting an even bigger bloom going on. So if you've been tracking the presentation, three different types of sargassum, and we argue, hypothesize, three different source regions for the different types of sargassum. So what are the factors that lead to these different distributions? We think might be the same factors that help us understand the nature of these blooms. And first question, or the first place people looked, is there must be nutrients. Something about the system has changed. There must be a new source of nutrients, or we have crossed a tipping point in nutrients to fuel and support these blooms. But at SEA, we figure, okay, nutrients are certainly a part of it, but that's not the whole story. And where we got this idea was just a serendipitous event. This was a trip a couple of years ago during COVID times. We had some students on board. We were sailing south through the Sargasso Sea. And in the Gulf Stream, we got lucky. All three types of sargassum came up in the same Newstein tone. I was like, this is gonna be perfect. I can teach the students how to identify the three different types. Put the sargassum in the aquarium, waiting till tomorrow to show them as part of class. That was gonna be classes. Learn how to identify the three types of sargassum. Well, we had a thunderstorm come through that night, and my aquarium got full of fresh water. And this is what it looked like then when I saw it in the morning. What had been beautiful looking sargassum, all three types, perfect specimens to teach the class. Now my sargassum, after a night being exposed to the rain, some of them were dying off. In particular, Natans 1 did not seem to like that freshwater rain event. Sargassum fluicans didn't do as well, and Natans 8 didn't do so bad. Looked just as good as when I had originally put it in. So that got us thinking that it must be temperature and salinity are going to be important factors. Nutrients, certainly, to fuel the growth, but temperature and salinity is going to play a role and perhaps help us understand the fluctuations of these blooms over time and the character of these blooms. So this work, fortunately, has been funded by the Doherty Charitable Foundation. So all of this experimental work is thanks to their support. So this is work that we have tried to do on the ship, growing sargassum in small jars on a ship, but it's hard to really maintain temperature and salinity at sea on a sailing ship. So eventually we had to move this work into more controlled settings and partnering again with a former colleague of mine who now teaches at Everett College. So we're making use of their greenhouse space there, and we're growing sargassum in small jars. We went and collected in the Gulf Stream, found all three types, different temperature baths, different salinities and nutrient concentrations in the different jars. So here are our temperature treatments here, our salinity treatments here. We didn't replicate every combination. So just across these temperatures and those salinities. And 10 replicates under each treatment condition for each type of sargassum. So 210 pieces of sargassum that we monitored every day and changed the water every day. And these are the growth conditions that we have, light dark cycle, and we kept the nutrients at a modest level rather than trying to enrich them. So we really wanted to see our temperature and salinity without an enrichment effect, masking any treatments. So there's our setup, keeping track and photographing and measuring each piece of sargassum every day for 3 weeks. And what we were looking at finally was then a net growth rate over time. And across our 3 forms of sargassum, across our different temperature treatments here, what we really found, the important finding here is, the initial sort of explanation was it's nutrients and warming temperatures. Or it's climate change. Warming temperatures are fueling this growth of sargassum. Well, maybe to a point, but what we found is that at our warmest temperature, all 3 species of sargassum showed a decline in their net growth rate. But with some nuance. And I really wish we had run 30 degrees Celsius, where I think we really would have found the sort of dividing line between the different species. But we see that Sargassum natans 8 doesn't do nearly as poorly as the other 2. Here's Sargassum fluicans, and it's Sargassum natans 1, the species that has the most northerly distribution up in the North Sargasso Sea during the wintertime. It can get pretty cold. Seems to be the least tolerant of these warm temperatures. they just completely disintegrated at our warmest temperature treatment. So we think there's a subtle difference in temperature tolerances at the high range. They all seem to be doing fairly well at our colder ranges. And for salinity, they all seem to struggle at these lower salinities, which it was surprising. We thought salinity was going to be another really big distinguishing feature in how they responded to growth here. So this was a surprising result. And in part, I think it's because we kept it, the baseline temperature here for our salinity treatments were 28 degrees. And I think that perhaps was a little bit too warm. We probably should have run those experiments at 26 degrees. We may have gotten a little bit better distinction between a salinity response. So overall, the growth across any of our salinity treatments here are pretty low overall. It may have been an effect of the high temperatures that we were running those experiments at. But we still saw then there's a lower threshold salinity response as well. So we think temperature and salinity are helping us understand, if we piece together what's going on in the equatorial Atlantic Ocean, how temperature and salinities have shifted across time. That might explain the nature of these blooms. So what is sargassum? Why should we care? Well, hopefully we have lots of reasons to care about sargassum now. Where is the Sargasso Sea? I still don't know if we've answered that question. Still ongoing. And I think it's because where you find sargassum is shifting, and it's continuing to change over time. What determines sargassum growth? Well, nutrients is certainly a factor, but we do have to pay attention to temperature and salinity going forward. And there are still, of course, lots of questions that remain. So if you're all interested in sargassum, perhaps you'll be the ones to answer some of those questions. All right, thank you. All right, who are here for some of the questions we have today?
Okay. Who has a question? Our student doesn't be here. Good question. Go for it, Brad.
Let's play this place science sargassum. I was just curious, why is sailboat for the research vessel? That's good.
Yeah, so wow, sailboat. That's the company I work for, is Sea Education Association. So this research has all been done in the context of delivering an academic program to college undergraduates. So this is a study abroad experience for those students. So yeah, Sea Education Association. Encourage you to check out any of the flyers and pamphlets I have about the organization. Encourage any of your friends that are still undergraduates to look up our program, check it out. They can help us continue our research on sargassum. So yeah, that was a platform that I work and teach for. But we teach students oceanography by conducting real world research. So they get their hands on doing that work. So that was the only platform I had available to me.
So do you think that the sargassum isn't like producing spores and new growth while it's just floating in the cycle, and it's more like for that one species in the Gulf of Mexico?
Yeah, a really good question, and some details I've left out. The planktonic forms of sargassum, there have been no evidence of receptacles or conceptacles or production of gametes. It is just growing vegetatively, breaking apart, and then if it's in a favorable environment, that piece will then branch and grow, continue to grow vegetatively. So it's certainly, if it's in the proper conditions, I'm sure that vegetative growth continues, depending upon as it moves into either favorable or unfavorable conditions. But certainly along that pathway, there is continued growth for sure. Yeah, back.
Have you ever, would you ever look at how big a patch necessarily needs to be to support such large fauna compared to just having smaller microorganisms and fauna?
There's some folks out of France that France seems to be funding a lot of research on sargassum, not United States, unfortunately. And that was one of many questions that they had on, this is a couple of years ago now. And Odie and others, Odie and others mapped. So they would get out there and map the size and scale of different patches. But they weren't necessarily then looking at the fauna afterwards. But it gives you a sense of the variety of sizes of sargassum and how it manifests itself, how it aggregates out at sea. We've done a little bit of that with just visual observations of at SCA, like when we see windrows, are we seeing seabirds? Are we seeing other fauna? And for the larger fauna, we're not seeing the larger fauna often enough to get anything statistically significant with more windrows, more seabirds, or anything like that. So that's another one of those questions that we still need to answer. And it's a good one. I think it was in the batch first.
Hey, John, clearly you're passionate about sargassum, it's very fascinating, but your sampling technique is so it's microplastics as well. Yes, that's probably all under talk, right? I mean, you must see a historical trend. Time that SEO started.
Yeah, maybe some folks remember Ed Carpenter.
Oh, yeah.
Ed Carpenter, when he was at Hui in the 70s, met Corwith Kramer. So the ship shown here is the Corwith Kramer, named after the founder of SEA. And Ed Carpenter and Corey talked, and it was Ed Carpenter who suggested, what should you do on board your ship? What can you measure? And he said plastics and sargassum with a new standard. And so I have to give credit to Ed Carpenter. He got SEA started in 1971 with our scientific sampling. So plastics, tar, sargassum, of course, then the plankton community as well. And SEA has the greatest archive of plastic distribution because of that since 1971. So you can find that paper, Lavender Law and others, 2011 in Science, showing our plastics distribution. But we have seen that last couple of decades, it was sort of holding steady. But then the last, like earlier decades, like this most recent decade has started to tick up again. And that has been a sort of surprising thing. We just sort of updated our sargassum and plastics data with the Sargasso Sea Commission. They want to update their promotion of establishing a high seas protected area. They were asking for our updated Newstein tow data. In this most recent decade, we've seen plastics start to tick up again, but it had been steady for about a decade for plastics.
So do you see any correlation with going back to the question about the size of the
Yeah, aggregations.
Does sea state have any effect in this? I mean, is that why they break apart or is some being chopped on by fish?
Yeah, you all have remembered laminar circulation and how that process will bring floating particles together at convergence zones, and then those areas are spread apart. These windrows are spread apart the divergent areas of your laminar circulation patterns. So under certain wind conditions, when the laminar circulation forms, you see those windrows develop, and usually then maybe occasionally, if there's enough sargassum, if there's a supply of sargassum, those windrows can be narrow and really hard to see, or get bigger and bigger. And then sometimes we'll start to see large patches within the windrows strung together. The largest patches, though, unusually, though there may be other boundary variables. The largest patches I have seen have always been under calm conditions, where it's not a windrow, but it's just like a football field of sargassum, just all together, but not strung out. And then maybe another nautical mile down the way, perhaps you come across another football field worth of sargassum. So the largest patches I've always seen calm conditions. So certainly it does influence how it aggregates. Oh, sorry.
Yeah, I've always heard about the sargassum sea in the context of like American eel research, and I'm curious if that's something like encountered.
For sure, and that was one of the pieces of information that the Sargasso Sea Commission was most interested in trying to create and establish this protected area. So you had to have that iconic species. So sargassum perhaps wasn't enough. Eels, you needed some vertebrate, I guess. Algae ain't cool enough, I don't get it. But you had to have that iconic eel species. And yeah, we get when we find sargassum, we find more eel larvae. So it seems to be, even though they're vertically migrators, as larval stages, they tend to seek it as a refuge. Yeah, the densities at nighttime are greater when sargassum is around. Yeah, sure.
So the fact that Nate has 8 has higher tolerance to high temperatures in fresh water makes sense if they do live in the tropics. And the tropics have kind of always been warm and fresh. So what has happened that has made it so much better for them over the past 10 years than it was before? And why was it bad for them for like 100 years between those observations?
I know, and I think that will just be one of those things where at the most we'll be able to conjecture, but nobody was there in the moment to see the origin of this event. Because I do think, you know, and again, this is some of the unpublished data you were asking about, these ideas of source regions. We've been remiss in actually telling this story, at least getting it out published. Of 3 types of sargassum in 3 different source regions. So that's still just, we need to get that paper out there. So, right, if you go back and read the fine details of Parr's work in 1939, the first one to sort of describe Nathan's age. He said he saw a rare sample in the Caribbean, and then he saw another rare sample off of Newfoundland, which perhaps must have come off of the Gulf Stream. So that was just sort of getting circulated around. So I think it's been around always. Why low abundance? I don't know. What has changed for that type of sargassum and why it's blooming? I mean, I think we'll get to points where we can hint at it once we understand precise temperature and salinity that it does best. And maybe go back historically and look like that happening in 2014 and 15. Maybe we'll be able to piece that story together. But I think it was one of those events where you had to be there at the time to know what started it off. And now I think we just have a seed population that is just able to maintain itself. You know, interesting conversation I was having with John the other day. Well, if you have this temperature limit, what is that going to mean for sargassum? Are we going to get to a point where the fluids are going to stop? Or I think maybe it's just going to shift where, and perhaps the seasonality will just shift to a more of a winter time rather than height of summertime for these nature of these blooms. So I think they're going to be around for a while, but it's a long way of saying I don't know. To your question, but it's a really interesting question for sure.
All right, thank you so much for this.